Hey 12 of you. So this is going to be our last lecture, our last content part of the course. So after we get done today, we're all done in the course, and now we're just prepping for our exam. Okay, now our exam for most of you is not going to be for a little while. Some of you are writing a little bit earlier, but you want to make sure that you do a lot of preparation for this exam. Keeping in mind some different things to do, I highly suggest going on, and especially in our last unit, watching some YouTube videos. I mean, it works for all of our units, whether you're talking about cellular respiration, or you're talking about macromolecules. The videos on there are excellent, and I in particularly love the crash course. I find the guy hilarious, very entertaining, a little bit easier to watch than maybe some of the other ones that may be a little bit dry. So our last lecture that we're going to go through today is on the Nephron. Now, I've already given out an assignment, and that assignment is optional. Okay, it's optional as to whether or not you want to do it. It's getting pretty close to exams. Some of you might already be in exams because we got kids all over the board. So I don't want to put this extra pressure on you. You got to get this thing done. But at least gives you if you decide not to do it, at least gives you kind of a framework to work by as to what type of questions is he going to ask. And the questions that I put in that assignment are more inquiry and application type questions. You can probably predict some of the knowledge-based questions, which is just pure regurgitation that I'm going to ask you. But it's sometimes difficult for students to try and predict, well, what kind of inquiry or what kind of application type questions are going to be asked. And that's what I kind of primarily put in the assignment. Okay, so if you have any questions about the assignment, let me know. I want you to submit it in that Dropbox like I put in the news feed. Okay, so let's go through our last lecture, which is going to be on the nephron, which is simply the functional unit of the excretory system. Okay, so the big thing we're going to be talking about is how we basically make urine. Now, the big thing I really want to clarify is when we're talking about the kidneys are the only ones that can actually clean the blood. Okay. Now, something like the liver can help detoxify things. So the liver may say your alcohol comes in your body, your liver is going to convert it to a less poisonous form. Okay. Now, when we want to remove that metabolite, or we want to move some of the waste products from that poison, the kidneys are the only ones that are able to do it. They're the only ones that are able to extract stuff out of our blood and then get it out of our body. Okay. The liver can't do that. So just something to think about. Okay. So this is what we're going to go through today, is a nephron. Okay. So when we look at the kidney, there's three main parts you need to know: the outer cortex, okay, which is the outer part; the medulla, which is kind of the inner part; and then the renal pelvis, which is right in here. Now, what we're going to be talking about is the nephron. The nephron is really connecting both the cortex, the outer part, and then the medulla, which is going to be the inner part. Okay? The nephrons will drain via the collecting duct into the renal pelvis, and then all the urine ends up here, goes down your ureter, and then will go right into your bladder. Okay? So we're going to be talking about a nephron. Now remember, we have millions and millions of these things inside our kidneys, but we're going to focus on just one because clearly we can't focus on all a million at the same time. So what is the kidney responsible for? It's for removing any waste products from the blood and ensuring the blood contains proper water, salt, and pH balance. The kidney does so with a functional unit called the nephron, and today that's what we're going to be talking about, is the nephron and how it basically turns our blood into urine. Okay, so it's going to go through four steps, and there's also four main parts. Okay, so let's go through this one at a time. The first thing we're going to talk about is glomerular. Sorry, I didn't pronounce that right. Filtration. Okay, so this is the idea: is you are going to have a renal artery, okay, or an efferent arterial, same kind of thing. You're going to have a large supply of blood coming in to your kidneys, basically, and then going into your nephron. So this is a blood supply. So this is like an artery going in, and you can see the artery is sealed, right? Because we're not going to be bleeding out all over the place. But right around the artery, we have this basically this little capsule that we call Bowman's capsule. And what's going to happen is most of the fluid inside the blood is going to leave and go inside this filtrate, okay, or this little capsule. So there's a couple things that are not going to go in, but you can look at this as really just a filter. We're bringing blood in and we're filtering out some things that we don't want to have in our kidneys and other things that we do want to have. Okay? So blood flows through the glomerulus, causing water to be dissolved in substances, leave the bloodstream, and enter Bowman's capsule. So here's Bowman's capsule, this yellow thing here, and this is the glomerulus, this series of little capillaries. Okay? Now the substance that entered Bowman's capsule left the blood, and it's considered the filtrate. So this is the filtrate right here, the stuff that has left the blood and gone into Bowman's capsule. Okay. Now, there's not that many differences between blood and filtrate. The key is that the filtrate is not going to contain any proteins. Okay, there's not going to be any proteins in the filtrate. And if there is proteins in the filtrate or in your urine, then you obviously have a kidney problem. Okay, so we don't want any proteins in our filtrate. We don't want things like red blood cells, which is also why the filtrate won't appear red. Blood appears red because it contains a number of red blood cells. But since the red blood cells can't go through the filter and get into Bowman's capsule, the filtrate is not red. Okay. If it was, I mean, we'd be peeing out red, which if you are peeing out red, definitely go see a doctor because that's not right. You're having red blood cells in your urine and you shouldn't have that. Okay. Some other things that can't enter are like our leukocytes, our white blood cells, hemoglobin, large different molecules that just can't get out of here. Okay. Now, two important factors allow this to happen. One, the highly porous capillaries of the glomerulus. So these capillaries right here are very porous. So their plasma membranes allow a lot of transfer out of the bloodstream and into Bowman's capsule. And the other one is the high blood pressure in the glomerulus. So when we pump in blood, we kind of put a lot of pressure inside each of these tubes, which will again drive liquids out of these tubes and into Bowman's capsule. Okay, so here's just kind of another picture of it. We got blood coming in. It's going to go through the glomerulus. Okay, and then the high pressure is going to push stuff out of the glomerulus and into Bowman's capsule. And then here we have another tube, and this tube is going to lead our nephron, and it's going to lead the filtrate all the way through this series of tubes. Okay, so 1,600 to 2,000 liters of blood gets filtered every day by your kidneys. Try and put that in perspective. That is a ton and a ton of blood. It's about 2,000 kilograms of blood. Okay, just massive amounts. Now, from those 2,000 liters, only about 180 liters of filtrate gets made. So we have a lot of blood going through here, and from all of that blood, the 2,000 liters that is going to go through, about 180 liters of this filtrate is going to be made. Okay. Now, 180 liters is, again, a ton, 180 kilograms. So just a ton of liquid. And we don't pee out anywhere near 180 liters of filtrate. If you're peeing out that much, I mean, clearly, again, go see a doctor. Okay? So we are going to reabsorb most of this filtrate, and we'll talk about that as we go through the nephron. Now, the filtrate and the blood are identical in composition with the exception of proteins and, some red blood cells and white blood cells. So when we look at this liquid that's inside here, the filtrate, and compare the liquid that's inside the artery, which is blood, there's not that many differences. Okay? You're both going to have sodium and chloride and potassium, and other small things like calcium, but you're not going to have the larger molecules that are in the blood, okay? AKA the proteins, some red blood cells, some of the bigger ones. Okay, so what we're going to do now is we're going to talk about how we reabsorb it, because we have 180 liters of filtrate going inside Bowman's capsule and going through these tubes, but we're not going to end up with 180 liters. We're going to end up with significantly less. So we're going to reabsorb a lot of this filtrate and then go right back into the bloodstream. Kind of seems like an inefficient system to put a whole bunch of filtrate and then reabsorb most of it, but they often say the kidneys are inefficient. Okay, so we're going to go to step 2. Now that we've passed the filtrate, so if I go back here, we're past the glomerulus filtrate, and now we're going into here, the proximal convoluted tube. And during the proximal convoluted tube, a lot of reabsorption takes place. 65% of the filtrate is reabsorbed. Okay, and this is including the loop of Henle, which we're going to get to. So reabsorption requires both active and passive transport, and we'll talk about when you use active, when you use passive. The first part we're going to talk about is the upper proximal tube, and this is prior to the loop of Henle. So if I go back to my diagram here, this is this right here. Okay, before we start dropping down at this loop of Henle, just in this area right here, we're going to explain what happens first. And in that area, you have a lot of reabsorption of glucose. About 100% of the glucose gets reabsorbed. So all the glucose is going back in. So you shouldn't really see glucose in your urine. Okay, sodium and potassium, chlorine and water can also get reabsorbed. Not all of it, but a lot of them are going to get reabsorbed here. Okay, so then we're going to go past the beginning of the proximal tube. So we're past this part, which we just did, and now we're starting to go in the loop of Henle. Okay, and we're going to start with the descending loop of Henle. And we call it the lower proximal tube or the descending loop of Henle. In the descending loop of Henle, the salty environment of the medulla. So here's the cortex, the outer part of the kidney, and here's the inner part of the kidney. And in the medulla, it's very salty. Now, you really want to remember that it is salty because it helps explain what's going to happen in all of this transfer of liquids. So we have a salty environment. Well, the salty environment is going to try and make itself isotonic with the liquids inside the tube. So if we remember from our unit 2, or passive transport, sorry, unit 1, we know that water is going to leave. Okay, water transfers a lot easier than salt will. So water is going to come out to try and make both of those solutions isotonic. So during the descending loop of Henle, a lot of water is going to come out. Okay, and it's being drawn out passively. We don't have to actively pump it out because this is really, really salty here. So the water is going to try and leave this environment in order to make this less salty. Okay? So the permeability of the salt of these cells right here is really, really high, and it's just going to pull the water out. Okay. Now, when we pull all this water out, any salt that's left inside here is going to remain. So when we get to the very bottom of the loop of Henle, it is extremely salty down here. So super, super salty down here. Okay. So we're going to go past the bottom loop. This really, really salty filtrate is now going to go up on the ascending loop of Henle. So now we're going to go up. Now the key with the ascending loop of Henle is we're going to stop water permeability right here. Okay? Water can almost transfer through this entire thing with the exception of this area right here, the ascending loop. Water can go back and forth to here, the descending, through here, and through here. But on this ascending loop of Henle, it's almost impermeable to water. Okay? So water pass through the filtrate to the blood. However, salt can and are actively pumped out of the filtrate. So if you notice at the very bottom loop of Henle, remember I talked about how salty this is. It's so salty that at the very beginning, it can passively leave. Okay? The medulla, or the outer medulla, is very salty. This environment, everything in white is really salty, but inside here is even more salty, so it will passively leave. Okay? Now, at a certain point, it's going to become isotonic, where the solution in here has the same salt concentration as the medulla. And when that happens, we're going to have to switch over from passive transport to active transport so we can go against the concentration gradient and actively pump out sodium chloride here. So now we're going to actively go against the medulla and pump out even more sodium chloride out of the ascending loop. Okay? And now that's going to drop the salt concentration of the filtrate as we keep pumping out salt. Okay? All right, so now most of our water and salt have been reabsorbed. Now, the key thing in this whole part that I've just talked about, this is going to happen to you regardless of whether you're dehydrated or hydrated. We don't have any we don't have any control over what happens at the beginning here. Okay? The control that we exhibit over water levels and pH happens in here, the distal tube. Okay? Sorry, I have the hiccups here. So, in the distal tube is where hormones are going to start acting on the distal tube and controlling how much water we should remove or how much water we should remain in, how much salt we should remove or how much salt we should remain in, how much things we should secrete. So this is where we can kind of control urine. And this is where, when you get dark urine or light urine, this is really what's controlling it. This part right here and then the collecting duct. Okay? Prior to that, in this loop of Henle and the proximal tubula, we don't have any hormonal control over how much gets reabsorbed or secreted. Okay? All of that control comes in right at the end. Okay? And that's when we have the distal convoluted tube and the collecting duct. So following the proximal tube, the loop of Henle, is the distal tube. Here, some reabsorption, salts and water, can still occur depending on the needs of the body. It's under hormonal control. If you need some more salts, or you have too many salts, you can start secreting or absorbing them. Okay? The other thing that's very interesting here, and it doesn't happen anywhere else, is you actually start secreting stuff from the bloodstream into the filtrate. So absorption is from the filtrate to the bloodstream. Secretion is from the bloodstream to the filtrate. So here in the bloodstream, we're actually going to start secreting some other things. We can secrete some H+ ions if we need to drop the pH of the blood. So here you're going to have H+ ions come from the blood and into the filtrate. Other things like other foreign solutes like medications or illicit drugs or steroids will also be secreted into the filtrate here and then go into urine. Okay? And then following the distal tube, the filtrate enters the final leg called the collecting duct. And this is where we have the most control over water reabsorption. So this is where your body is going to look at, are you dehydrated or are you overly hydrated or are you perfect? And then control how much water gets reabsorbed during this final phase. Okay? So the collecting duct is under control of ADH, antidiuretic hormone, okay, which is again released by the pituitary, which can either increase or decrease water reabsorption in this final leg. If the person is dehydrated, the collecting duct will upregulate the reabsorption of water and urine will be dark. So what will happen is it'll open, if you're dehydrated, ADH will act on the walls of the collecting duct and open up a whole bunch of channels, water channels, for water to come out. Now, water can enter already through the plasma membrane, but they're also going to open up some aquaporins or channels for water that just kind of flood out water. So you reabsorb all of it, and then your urine becomes really, really dark because all of your water went back to your bloodstream. Now, if you're overly hydrated or you have enough water or excess amounts of water, you don't want any water to leave the filtrate and go into your bloodstream because your blood is already very thin or it's got lots of water in it. So what we're going to do is we're going to leave all the water inside this tube. We're not going to reabsorb any of it, and we're going to go right down to our bladder, and you're going to have, more of a dilute type of urine. Okay, so the last thing I'd like you to do, this video is the crash course. We already watched it. I posted another video in the news feed, which I suggest watching, and this is a very good activity that I highly suggest doing. Okay, not something that you're handing in, but it's more of a practice for making sure you understand the nephron and where reabsorption is happening, where passive, where active is, and where secretion is. Okay, so you can use a diagram from Google Images. So just under Google Images, type in the nephron. And I'd like you to label, where we're doing active reabsorption, we're doing passive reabsorption, and then where we're doing secretion. Okay, you should also know, what's coming in and out during the different areas of the nephron. Okay. All right, and there's just a picture of it. So that's pretty much it. Okay. Now we are done the course. I know it's been a long course, but you reflect back and think of how much you've learned this year. Everything all the way back from macromolecules and building a cell, how we test for them. So we need to start studying now. Okay. I'd like you to finish up that activity at the end and then prepping for our exam. Now I'm going to give kind of an overview. I'm not going to give you a review, but I'm going to kind of do the 4U course in a flash in another kind of video series that I have for you. Okay. So if you have any questions or concerns, make sure you email me. Again, that assignment that I posted, if you hand it back into me by Monday, it has to be Monday, I will mark it and I will add it to your present mark. Okay. If you don't hand it in, you're not punished at all. You don't get a 0 on it. You just get a no mark. It's like you never, it doesn't help you. It doesn't hurt you. But what that means is that the mark that I've already emailed to you is the mark that you're going